Assalamualaikum and good evening. For this session today, you are with me, Shamila Azman from Water and Environmental Engineering School of Civil Engineering, UTM. Welcome to UTM Engineering Distinguished Lecture Series for today. Alhamdulillah, today we will continue with lecture series number 21, brought to you live from Faculty of Engineering, University Technology, Malaysia. For all the viewers from inside and outside UTM, and everywhere around the globe, please like our FB page. Please do share our FB link with your friends and contacts. Indeed, it is a great honor for me to host our honorable guest speaker today, distinguished Professor Dr. Yoshihisa Shimizu from Graduate School of Engineering, Kyoto University, Japan. Prof. Shimizu is a very well-known researcher worldwide, and he is the editor for Environmental Science and Technology. He is also the Japanese coordinator for Asian-based and risk-oriented integrated watershed management. Malaysian researchers have benefited a lot under this program, which is also known as JSPS Asian Core Program. We also have student exchange with Kyoto University. Without further ado, I would like to invite Professor Mohammed Rafi Abdul Kadir, Dean, Faculty of Engineering, University Technology Malaysia, to introduce our guest today. Over to you, Prof. Rafiq.
Thank you, Dr. Shamila. Assalamualaikum warahmatullahi wabarakatuh. Hello and welcome everyone. Welcome to our 21st UTM Engineering Distinguished Lecture Series. My name is Mohammed Rafiq and I am the Dean of Engineering, University Technology Malaysia. Today, it is my utmost pleasure to welcome Professor Yoshihisa Shimizu from Kyoto University, Japan. A bit about our presenter today. Yoshihisa Shimizu is a full professor since 2007 at Kyoto University, Japan. He obtained his PhD in 1990 from the University of Texas at Austin. His field of interest is environmental engineering, especially in one Asian-based and risk-oriented integrated watershed management with hydrology, water quality, environmental risk, and governance. Number 2, characterization of natural organic matter and elucidation of its role and function in the aquatic environment. Number 3, recovery of inorganic nutrients by urine diversion, biotoilets, and its benefit assessment. And number 4, development of physico-chemical method to remove organic micro-pollutants in the aquatic environment. Yoshihisa Shimizu is a member of 2 global coalitions of researchers, the International Educational and Research Activities in Global Environmental Studies, and the Global Center for Education and Research on Human Security Engineering for Asian Megacities. He is the recipient of the Stockholm Junior Water Prize in 2014 and the Japan Water Prize in 2012. He is the president of the Society of Environmental Instrumentation Control and Automation since 2012 and the editor for the journal Environmental Science and Technology. So that is a biography of our speaker. Here now is Professor Yoshihisa Shimizu from Kyoto University with a talk entitled Simulation of Hydrology and Water Quality for Integrated Watershed Management. Professor Yoshihisa Shimizu, over to you.
Thank you very much for your kind introduction, and it's my great honor to be a speaker here in this distinguished lecture series. And my name is Yoshihisa Shimizu, and Yoshihisa is my first name, and Shimizu is my last name. Sometimes in Japan, we use Chinese character to express our names, and in that sense, Shimizu, for Shimizu, my last name, we use 2 Chinese characters. The meaning of that is clean water. Maybe that's the reason why I am here in this business. And actually, I have been to Malaysia, including Johor, Kuala Lumpur, and other places, more than 50 times so far. But unfortunately, I do not have any opportunity to go to Malaysia this year, but soon I'd like to visit there again. And the topic today I'm going to talk is the simulation of hydrology and water quality for integrated watershed management. And today I am going to talk about the dioxins, the world's most toxic organic chemicals. And the field of my study is, in this case, Lake Biwa. This is the biggest freshwater lake in Japan. And more than 15 million people are getting water for agriculture, for industry, and for drinking from Lake Biwa. 15 million means that more than one-tenth of Japanese population. And the final goal of this research, which I'm going to talk about today, is we would like to do the virtual simulation, which means we don't have to be there. We don't have to install any wastewater treatment plant. We don't have to install any water treatment plant. Even we don't have to construct any building, road, and we don't have to destroy our forest. Actually, we can do these things in computer that we call virtual simulation using famous geographical information system, GIS. And today the focus is on microorganic pollutant, and especially the focus is dioxin. Again, the most toxic organic chemicals in the world right now. And in order to find out the movement or fate of dioxin microorganic pollutant, we have to know the hydrology in the watershed. And even though I am going to talk about or focus today is on dioxin, but the simulation is not only dioxin, but also for other pollutants, including BOD, COD, suspended solid, or not pollutant, but pH, and so on, nitrogen and phosphorus. And today I would like to use the dioxin as a marker to verify our simulation or to estimate the future movement and fate of some other pollutant, including inorganics and organics. So we would like to use in this research dioxin as a marker. Even though the dioxin is the most toxic organic chemicals in the world, at the same time, the dioxin is inert, very difficult to be degraded in the environment. That's the reason why we can use dioxin as a marker for our simulation. And fate and movement, when we think of the dioxin, usually we call dioxin is hydrophobic microorganic pollutant. Hydrophobic means the dioxins are a kind of hate to the water. And microorganic pollutant concentration in the environment is very small, but toxicity is very high. Since the dioxin is hydrophobic, so the dioxin is attached or adsorbed, or sometimes we say sorbed, to soil and suspended solid, especially the organics of soil and suspended solid. And with the soil and suspended solid, the dioxin are moving around in the environment. So we need to estimate the movement and fate of dioxin at the same time again. By verifying our simulation by using dioxin, we can come up with the simulation of any other organic pollutant in the watershed. And today, I would like to talk about these things. First of all, we have to have samples from surface soil or surface plant, sediment, and waters in rivers and in the lake and also in the atmosphere. Actually, for this research, we took more than 1,000 samples in different environments, And then we have to determine the dioxin concentration of these samples. And the official method when we started this research in Japan was so-called GCMSMS, gas chromatography and mass spectrometer in series. The instrument itself was and still is too expensive, and for one measurement, for one analysis, it normally takes more than 2 weeks. And the running cost of the analysis itself is about 1,000 US dollars per sample. Since we have too many samples, more than 1,000, it is very difficult for us to measure all the samples by these official methods by GCMSMS. Instead, we use so-called bioassay. This bioassay has a special name which is called KERAC. I will briefly explain the system of KERAC itself. And then having the value or having the concentration of dioxins in several environmental samples, we would like to find out the relevant characteristics of surface soil and dioxin concentration. We had found a very interesting result from this, so I would like to explain that. And then with these measurements, and at the same time, the simulation of hydrology and water quality, like suspended solid, movement of soil and suspended solid in the watershed, and also the movement of dioxin, we would like to estimate the behavior and distribution of dioxin in the watershed. And especially in Japan, when we talk to the ordinary people, and when we ask them, do you know the origin of dioxin? First, they say the incineration. Since the land space is limited in Japan, and also we have to have the kind of safe for the treatment of the solid waste. Normally, we use incineration for the treatment of municipal solid waste. And due to the incineration, the dioxin is more or less released to the atmosphere through the stacks. And that dioxin released from the incineration plant through the stacks eventually go to the forest, paddy field, agriculture field, or urban area or water area like rivers and lakes. But at the same time, in Japan, we applied, not incidentally, accidentally, the chemicals, agricultural chemicals, to our paddy field. But in that agricultural chemicals, unfortunately, we synthesized, unintentionally synthesized the dioxins in these agricultural chemicals. So the contribution of the incineration plant is very high, but at the same time, contribution by the agricultural chemicals is still high. Talking about agricultural chemical application in Japan, x-axis is the year from 1960 to around 2000, year of 2000, and vertical axis, y-axis is the consumption of some agricultural chemicals. The red line shows the consumption of PCP, pentachlorophenol, and the primary product or primary agricultural chemical was pentachlorophenol. But due to the production of pentachlorophenol, during the production of pentachlorophenol, unfortunately, this amount, 400 kilograms of dioxin, was included in pentachlorophenol. And these dioxin are sprayed in agriculture field in Japan. And around 1970s, we noticed that in PCP, these dioxins were included, and we banned, Japanese government banned the production and the use of pentachlorophenol. And instead, we started to use CNP, this is chloronitrogen. But again, this chloronitrogen included dioxin, about 200 kilograms. So the total amount dioxin sprayed in Japan into the agriculture field is more or less 600 kilograms. This is enormous amount. And in Vietnam, during the war, the military of United States sprayed some chemicals which contained dioxin. The total amount of that sprayed was around 400 kilograms. So the amount in Japan, or amount the dioxin sprayed in Japan, was so high. And some of you might know the location of Lake Biwa. The location is around here. This is the capital city of Japan, Tokyo, and we have Osaka and Kyoto, the very historical city where I am living is around here. And Lake Biwa is the shape is something like that, and the watershed is something like that. And the distance from south to north of Lake Biwa is about 60 kilometers, 60 kilometers. And surface area is 387 square kilometers. And 14 million people are getting water, including Kyoto and Osaka. The people living there are getting water from Lake Biwa. So once this lake is contaminated by different or some chemicals or some pollutant, it is not good for us. And this is the topography of Lake Biwa. And there are more than 300 rivers flow into Lake Biwa, and only two rivers flow out from Lake Biwa. One is natural river, eventually connected to Yodo River to Osaka from here. Another one is artificial canal to Kyoto City, which was constructed about 150 years ago. And several watersheds are there since more than 300 rivers flows in. But three major watersheds, one is called Yasu River watershed, and the watershed, the land use in that watershed, the land use is basically for agriculture, paddy field. And another one is very north, Ane River watershed. This is mountainous area. The land use is basically mountains and forest. And Ado River watershed, partially the watershed here in Ado River, the land is partially used for agriculture and partially forest. So first we sampled the surface soil from Yasu River watershed, Ane River watershed, and Ado River watershed. By the way, my Research Center for Environmental Quality Management, is located here, not in Kyoto City. The Lake Biwa watershed is actually in Shiga Prefecture, not in Kyoto Prefecture, and my research facility is located very south of Lake Biwa, Lake shore of Lake Biwa. And this is a sampling picture of some sampling methodology. And for surface soil, my students are getting the surface soil by using stainless steel, and container must be either stainless steel or polycarbonate. If you use other plastics, then the dioxin, the concentration is very small, might be attached to the inner surface of these containers. So we want to avoid these effects. That's the reason why we used inorganic stainless steel shovels and either polycarbonate or aluminum or stainless steel container for the collection of the samples. And sometimes we need to obtain the samples from the sediment or from the river, sediment of the rivers or sediment of the Lake Biwa. And in order to obtain the sediment, we use these cylinders. And with attach some heavy weight, we just draw these cylinder into the bottom, to the bottom of the rivers and to the bottom of the lake. And once it's hot, once it's hit to the sediment, due to the weight, it is penetrated into the sediment. And then after the penetration, without the cap, we slowly brings this cylinder to the surface. In case of lake, we use the boat. In case of rivers, we just go there. And then as soon as it hit the surface, it comes up to the surface. By hand, we just cap at the bottom. But sometimes we lose all the sediment due to the gravity. If the particle size distribution is so big, we might lose. The possibility of losing the sediment is very high. So we had to try so many times to collect the sediment. And here is some colors and textures you can see from the sediment, from the surface soil forest, from the paddy field, and from the dry farmland. And the colors are totally different, and particle size distribution and some characteristics are totally different. It is very much depending on the surface soil, on the location. And this is the bioassay we used instead of using expensive official method in Japan. And right now, this bioassay became unofficial method of Japan. And actually, this bioassay is very cheap. I said for GCMSMS, we have to spend about 1,000 US dollars per sample. But in case of this bioassay, the cost for the measurement, cost for the analysis is just $1 per sample. And also the duration of time, the time required for the analysis is more than 1 week, less than 1 week. But even though I say less than 1 week, we can measure the concentration of dioxin, more than 100 samples of concentration of dioxins of more than 100 samples within a week. So the time duration and the cost are totally different. Without having this method, we could not come up with the result of this research. Briefly, let me explain the detail of the mechanism. And first of all, we used the yeast cell, a cell of yeast, and we mixed the yeast cell with a solution which contained dioxins or PCBs or furans, dioxins. First, these dioxins in the sample penetrate through the cell membrane. And once the dioxin get into the cell, the dioxin is attached to so-called receptor. And with the attachment, this receptor migrate into the nucleus. And then in the nucleus, it is attached to the specific region of DNA. And the downstream of DNA, we manipulate it to produce so-called luciferase. This is an enzyme. And so once it is attached, dioxin is attached with the receptor to DNA, the luciferase automatically, this enzyme automatically produced. And this luciferase itself is enzymes used by fireflies, which can release some light, which means if the sample, the concentration of dioxin is high, which means the intensity, light intensity released from the cell itself is high. So with the relationship of concentration of dioxin and light intensity, we can come up with the concentration of dioxin in the samples. And even though we use bioassay, so-called KRAX, still for some samples, not all for 100, all of 1,000 samples, we have to make comparison between our official method and our bioassay. There are some distribution here, but still R square value is more than 0.8, which is not so bad. So we decided to use basically this bioassay, and for some samples, we decided to use GCMSMS. And this is some result. And we took the surface soil from 3 major watersheds in Lake Biwa watershed. These are Yasu River, Ado River, and Ane River. And these are average concentration of the forest soil, the average concentration of dioxin from the forest soil. And these are average concentration of dioxin in the paddy field. And if you make comparison between the concentration of forest and paddies, the concentration of paddies are much higher than the concentration in forest. The origin of dioxin in the forest is considered to be the chimney or stacks of the incineration plant. And in case of paddies, addition to the incineration plant, we have to think of the direct application by agricultural chemicals to the paddy field. So this is the reason why the concentration of dioxin in the paddy field are a little bit or much higher than the forest. That's of forest. And then we also measure the concentration of the dioxin in sediment in river and lake. These are average values. And if you make comparison, you can clearly see that concentration in this sediment are lower than the concentration in the surface soil, which means even though the dioxin are released from the stacks of the incineration plant for so many years, and dioxins are applied to the agricultural chemical directly so many years ago, but still these dioxins are basically remaining in the surface soil, and gradually, very small amount are gradually released to the rivers and lakes. And then we just focused on the surface soil. And on the surface soil, first, these are concentration of dioxins in the paddy field, surface soil in the paddy field. And x-axis is organic carbon content in surface soil. There is no relationship, almost no relationship between organic carbon content and dioxin concentration in the paddy field. But if you look at the forest soil, this is a forest soil from Yasu River, the relationship between organic carbon content in forest soil and dioxin concentration is very much or more or less linear. R square value is 0.86. This is one of the major watersheds, Yasu River watershed. And then Ado River watershed, the slope, still we have linear relationship, but the slope is a little bit smaller than the concentration of dioxin Yasu River watershed. And the third watershed, this is Ane River watershed. The slope of the relationship is very much similar to that of Ado River watershed. And the difference of these slopes are about 1.7 times. Please remember this value, 1.7. And then, since the dioxins in the forest soils are coming from basically the chimney of incineration plant, what we did was this is a map of Lake Biwa and Lake Biwa watershed, and this is Yasu River watershed, and Ado River watershed, and Ane River watershed is here. And these red dot incineration plant around Lake Biwa watershed. The number of incineration plant in the past and even now, the number is very big in Japan. And the total number of the incineration plant, including big, medium, and small, the total number is 724. And from these incineration plant, lot of dioxin, sometimes small concentration of dioxin have been released. So what we thought was we made up so-called atmospheric dispersion and deposition model. This is very simple. From one incineration plant, of course, the amount of municipal solid waste incinerated every day are different. Sometimes the incineration plant has a period of on and off. Sometimes big incineration plant continuously incinerate the municipal solid waste. And also we have the direction of wind. If the wind is blowing from left to right, the dioxin is sprayed into the atmosphere like that. It, of course, the distance is depending on the wind speed. And also, if the wind is coming from right to left, dioxins go to the left-hand side. And we have the rainy season, we have rainy period, we have clear sky period. So we included these information into the dispersion model and deposition model for each incineration plant more than 700. And then what we did was we superimpose. If there are two incineration plants, just we superimpose the simulation for more than 700 incineration plants. We superimpose more than 700 of them in our simulation model. For last 50 years, we simulated. And this is, again, Lake Biwa watershed and Lake Biwa. And these colors are the amount of dioxin deposited on the surface. Dark color means high deposition, and pale color means low deposition. Around Yasu River watershed, concentration seems very high. Ado River, Ane River, and Ado River in these watershed concentrations on the surface are not so high. And then in those three watersheds, we calculated the concentration of dioxin per square kilometers of surface soil, 0.093, 0.121, and 0.220. And remember, the slope of the previous graph in Yasu River is 1.7 times higher. And the slopes of the previous graph in Ane River and other rivers are almost similar. But x-axis in the previous graph is organic carbon content of the soil. So then we recalculated the value or the concentration of dioxin in the surface soil per gram of organic carbon. Here, the value from Ado River and Ano River are very much same. And in Yasu River, this value 1080 is about 1.7 times higher. So we concluded that the dioxin in the forest are basically coming from the atmosphere. The origin of that is incineration plant. There are so many in Japan. And in agriculture field, addition to the atmospheric disposition, we have the direct application of agriculture chemicals. And then we estimated the dioxin concentration of the surface soil. In the forest, in these three watersheds, these amount of dioxins are there. And then from the forest, in the forest of Lake Biwa Basin, we have about 3 kilograms of dioxins. And in addition to that, we have paddy field. The values of dioxins are something like that. And in paddy field, 14 kilograms of dioxins are there. So the dioxin from the incineration plant is about 3 kilograms dioxin from the direct application is more than 14 kilograms. Total is about 17.5 kilograms of dioxin are still there in the surface of Lake Biwa watershed. And now we know the concentration of dioxin in the surface soil. But with the rain, with the surface runoff, these dioxins are transferred to the rivers, into the water phase, and also in the sediment of the river. And then it moved to the Lake Biwa. It moved to the water of Lake Biwa and moved to the sediment of Lake Biwa and also moving out from Lake Biwa. So in order to simulate that, we used GIS, Geographical Information System, and we came up with Watershed Simulation model. And first we have pollutant, we have the information of land use, we have the information of soil type and weather information, meteorological information, and elevation, and so on. And then we can come up, or we can simulate the hydrology, flow rate of the water in the rivers, and also the water quality, in this case, the dioxin concentration. And in one way, in one side, the simulation we did, and on the other side, even though we knew the concentration of dioxin in the surface soil, then next stage, we have to know the concentration of dioxin in rivers, river waters. Even though we say river waters, basically these dioxin characteristics is hydrophobic, so it's basically attached to the suspended solid particulate matter in river. But some portion of dioxin is freely dissolved in river water. So we have to measure, even though one side we have been doing simulation, but to verify the simulation, we have to measure the next stage. We have to measure the concentration of dioxin in river water, including the dioxin attached to the particulate matter in the river water. So we use this kind of device. Again, all of them are made from stainless steel. And first, we pumping out the river water, and we pumped the river water through the flow meters. And then after the flow meter, the river water went to this device. And inside of this silver device, we have this kind of things inside. And big cylinder, the diameter is about 30 centimeters, and small 3 cylinders, diameter is about 10 centimeters each. First, river water goes through this big cylinder, and in this cylinder, we have filtration paper. And by using the filtration paper, we can remove or we can separate the particulate matter. Particulate matter is going to be deposited on the surface of filter paper. And after that, after removing of the particulate matter, the water goes through 1, 2, 3 cylinders in series. And inside of this cylinder, each has polyurethane foam. This is a kind of organics. So the dioxin dissolved in water phase can be absorbed in this organics. So after doing this kind of separation, first we remove extracted dioxin from the particulate matter, and we also the dissolved dioxin attached to this polyurethane foam by using some organic solvent. And then we measure the concentration of dioxin in particulate matter in river water and the concentration of dioxin in dissolved in river waters. And then also we need to verify our simulation in terms of flow hydrology and in terms of the suspended solid concentration continuously. And this is a device to measure the suspended solid or turbidity. We just use this one in the river. We just throw this one into the river. And this is the cylinder, stainless steel again. And one side we have the light is coming out. And on this side, we have the detector of the light. And we have been put or set this device into the river for more than 1 year. So for more than 1 year, on both surface, glass surface, there are windows on both sides. The surface can become dirty by algae sometimes and by suspended solid or by the accumulation of or precipitation of some dissolved matter. So we have the window, so-called window wipers, to clean up periodically on both sides. And the turbidity signal from that previous device is coming to this kind of device. And this is a case of the cigarette, one pack of the cigarette. And this device itself is not so big. And every 30 minutes, the turbidity is coming to our cellular phone. And here's the label. And we have solar panels since we need electricity. You can see some silver chain there. So that cylinder device is synced to the bottom or to the middle of the river, and the signal is coming periodically. Now, we know the turbidity, concentration of particulate matter, suspended solid in the river. And at the same time, we have to simulate the hydrology flow rate. Here, the blue line from January to December in a year, in Yasu River, the blue line is a precipitation, the intensity of rainfall. And these two lines from the bottom, one is estimated by simulation flow rate. One is measured flow rate in Yasu River Basin. One is green and one is red. Somebody said very difficult to see. So I replot the flow rate, and the x-axis is simulation, y-axis is measured value of hourly flow, and monthly flow, simulation, and measured value. From the R-square values, our simulation on the flow is not so bad. And then, by knowing the flow rate, by knowing the precipitation, we estimated particulate matter. And also, by that device, which I previously showed, we measure the turbidity or the concentration of particulate matter. And again, estimated value and measured value are very good match. And then, since we know the concentration of dioxin in the surface soil, and we know the flow rate, and then we know the concentration of suspended solid, that this is a runoff into the river from the surface soil, then we can come up with a concentration of dioxin in the rivers. Something like that. Suspended solid, estimation of suspended solid, and the estimation of dioxin in the rivers from January to December. And in Japan, we have paddy field, and we have rainy days and clear days. And when we have fine weather without any rain, more than 90% of dioxin run off into the river by the attachment of suspended solid. The movement of suspended solid accelerates the movement of dioxin. The only dissolved percentage is less than 10%. And under the rainy weather, we have 77% of dioxin is run off. And also, addition to that, even fine weather, some dioxin or some suspended solid are released into the river, but we have the paddy field, and in spring, we have so-called ploughing period. And in this ploughing period, first we store so much of water in paddy field and gradually release the water in the paddy field to the river. Only about 2 weeks in a year in spring. But with that activation or activity, with that activity, dioxin released into the river is about 20%, which is very, very big. And both rainy days and release by ploughing period, in ploughing period, most of the dioxins are attached to the suspended solid. It is also true, again, in the rainy days. Now, we know the concentration of dioxin in surface soil. We know the flow rate of the rivers. We know the movement of dioxin in the rivers. And then the next thing is we need to know the concentration of dioxin in the lake. So we selected several places in the lake and we obtained the samples. And especially here at other point, we just obtain the sediment from the surface. We use so-called Ekman bird, that's a grab sample. But at this one point, the depth of the water is about 70 meters. And we use so-called core samplers. Remember, I talked about the cylinder like this, but at this point, we use core sampler, a cylinder. The diameter is 20 centimeter, a little bit bigger than the normal. And then we just collected the samples, the sediment sample from here. And on the boat, we just sealed at the bottom and we made it upside down. And from the top, we sliced the sediment every 1 centimeter. So we obtained 27 slices. So the depth, the sediment we obtained is about 27 centimeters. And then in that core samples, of course, at each slice, each 1 centimeters, we measure the concentration of dioxin. And at the same time, we measure the concentration of lead 210. This is the isotope. By measuring this isotope, we can come up with the year the sediment deposited from 1920 to 2000. And the distribution of dioxin is something like this. This is a measured value. So in 1920s, 1940s, of course, we did not have any incineration plant, but still ordinary people just incinerate some garbage in their house yard and so on. So the still small concentration of dioxin is released and deposited in the sediment of Lake Biwa. And then 1960s, 70s, we started to use incineration plant, and also we applied agricultural chemicals, and gradually the deposition is getting bigger. And 1970s and 80s, we banned application of agricultural chemicals to the paddy field, and gradually the concentration in the sediment decreased. And around year of 2000, we established a kind of stricter regulation for the release of dioxin from the stacks of the incineration plant. And nowadays, the concentration released from the stacks of the incineration plant is very, very small. So the trend is something like that. This is the dioxin consumed in the paddy field, and the lag time from the application place to the sediment of the lake viewer is about 5 or 10 years. And in the bottom of lake viewer, we obtain the grab sample, surface 10 centimeters, and beneath of that again 10 centimeters, and the distribution of dioxin in the sediment total is about something like this. About less than 1 kilogram of dioxins are existing at the bottom of Lake Viewer. Very small amount. If you think of 14 kilograms of dioxins are still remained in the paddy field. And this is the mass distribution of dioxins in atmosphere. From atmosphere, some deposition is going on, and from the surface to the atmosphere, from the incineration plant to the atmosphere, certain amount of dioxin is released again. And in forest, in Lake View watershed, this amount of dioxins are there. And chemical application is 49 kilograms. And still 14 kilograms of dioxins are remaining in the paddy field. And gradually it is released from the rivers, very small amount. And in the sediment of Lake View, less than 1 kilogram of dioxin are there. And then, of course, gradually these sediment and water are released into two exits. One is natural river and one is artificial canal from Lake Biwa. But still, most of the dioxins are staying and released in the sediment and in the water of Lake Biwa. And by consumption of the fish or some shell, the dioxin is actually recirculated into the watershed from the lakes into the watershed. And here we can come up with the simulation and we can come up with a real measurement of dioxins. And again, it might be redundant. We could do that for dioxin. And of course, we can do the same thing for other pollutant. And by the simulation, again, this can be applied as virtual. We can move some wastewater treatment plant actually located here instead of here. We moved here, or we moved any other places. And in the computer, of course, by the movement, we can come up with the proper or better place of the wastewater treatment plant, for example, in the watershed. And also we can come up with the better process, what kind of unit process are required for that wastewater treatment plant. Of course, on water treatment plant, same thing. Incineration plant, same thing. Environmental facility, same thing. And here is the watershed. Water are recirculated, and sometimes pollutants are moving out from the watershed through the river to the downstream. But sometimes the pollutants are coming back. So watershed is very important to think of. And even though I said just a watershed, surface, river and lakes and some plant, I didn't mention the concentration of the dioxin in the plant, but still it is there. And at the same time, we have to have the information of the underground through the infiltration of the rainwater to the groundwater aquifer. And of course, our simulation model include this kind of information. And here is one example. This is an example in very famous river in Malaysia. This is an example of Selangor River in Malaysia, and from January to December. And we simulated the water flow in Selangor River in the year of 2050. This simulation includes the climate effect. And in the orange line here, there are some upside down. You have rainy season or wet season, and you have dry season in Malaysia. This orange line said in dry season, since the climate change and also the population grow and the water consumption grow, then in your Serangoon River in summer, it's gonna be drying up. So with the simulation, we suggested to use groundwater, not only surface water. We suggested to use groundwater year round. Every month, please take same amount of water from the ground. In that case, your water in the summer or dry season is coming back. That is the blue line coming back a little bit. But in wet season, since in the future, the rain in the future going up, then you might have in wet season, you might have flood. The blue line is much bigger here. So eventually we suggested only dry season, just use the groundwater. And in wet season, don't pump up the groundwater. Then in that case, you can conserve the groundwater at the same time. The flow rate in dry weather, dry season is much higher, and flow rate in wet season is much smaller. So you can avoid the drought, you can avoid the flood. So this is a very simple example in Serangoon River Basin in Malaysia, in Kuala Lumpur. And we have to think of the hydrology, of course. This is the hydrology, water quality, and some emerging pollutant. In that case, we have to think of environmental risk. And at the same time, we have to think of the governance. Very close to my final slide, for the integrated watershed management, we need science and technology institute, people's participation, finance, budget, and information and policy. But most importantly, we need to think of by ourselves, and we have to think we want to have cleaner environment in the future. This is a problem of our heart. So in that sense, sometimes I said we need hardware, and for the simulation, we need software, and at the same time, the most importantly, we need so-called hardware. And this is the end of my slide. Telemachusy, thank you very much.
Thank you very much, Prof. Shimizu,
Thank you.
for
A
your
little bit
information.
longer.
The question and answer session. On behalf of, okay, I have a question to ask. If we use for the sediment sampling, right?
Yes.
Prof use the core sampler.
Yeah.
What if we use the Ekman dredge sampler, the one that scoop up the sediment sample?
Yeah, that is a grab sampling methodology. So you can just get the more or less bulk sediment from the surface. So, but in that case, you cannot analyze the deposition in the air. So you can just measure the concentration in the sediment. That's it. But still, the value or the analysis is very important.
Okay, so we can still measure on the dioxin on the surface, right?
Of course, of course.
And another question is about the measurement of turbidity together.
Yes, yes.
What is the relation with dioxin then?
Okay, if the concentration of suspended solid particulate matter, or in other words, turbidity itself, if that value is higher, the dioxin concentration is higher. Since most of the dioxins, more than 90% of dioxins, are attached to these particulate matter.
Okay. All right. So we're still waiting for the questions to come. Okay, that's a question from Dr. Amirul Abu Bakar. I will read first, Prof. The dioxin concentration is found as highest in the dissolved condition during the fine weather as compared to rainy weather and during plowing. I'm very keen to know the explanation behind this finding.
Okay, let me go back to the slide. Okay, this is it. Okay, in a year, the most dioxins are coming out in rainy days, more than 70%. For the in the ploughing period, about 18% of dioxin, this is only a week or 2 weeks in a year, is coming out. And in fine weather, very small amount of dioxin is coming out from the land surface. But in each period, most of the dioxins are attached to the particulate matter, suspended solid. So the dissolved amount is very small. And the reason why in fine weather, the reason why we have 9.2% in dissolved, and in these conditions, the amount in dissolved concentration is very small, is the concentration of suspended solid here in fine weather is very small. That is the reason why. So the most of the dioxin, or the amount of dioxin dissolved in the fine weather, is small. Amount of dioxin dissolved in fine weather is relatively large because the concentration of the particulate matter or suspended solids are relatively small. That is the reason.
Okay, thank you, Prof. We are still waiting if there's another question, but our time has exceeded 1 hour, 2 minutes. I think on behalf of the organizer, we appreciate you making time to be with us today. Thank you again, Prof. Shimizu. We hope to continue our collaboration with you in the future.
Yes.
Thank you very much.
Thank
To
you very
all
much.
the viewers, do like, comment, share, and subscribe to our FB and YouTube channel. Thank you very much, all, and have a nice day.